last speaker. I think Shailja is not here. So I would like to invite Dr. Sabyasachi Senapati, is Assistant Professor in Department of Human Genetics and Molecular Medicines, School of Health Sciences, Central University of Punjab. Yeah. So it's area of interest in genetics and genomic of common and rare diseases. He did his PhD from Delhi University and visiting PhD fellow in Netherlands, and currently he is a assistant professor over there. So I would like to invite Professor Sabyasachi.
For the kind introduction. So I'm very happy to present here, and I'm thankful to ILCD to invite me in this conference to deliver a short talk on the work that we have done in last 3 to 4 years. My group is very young. I have just joined in 6 years back, and this group is working on genetics of celiac disease, which is an autoimmune disease of small intestine, duodenum, and the main effect is basically the venous atrophy, and induced by a protein called as gluten that gives glue kind of property of wheat, grain and barley, and this is acts as a the principal antigen. And what it does actually in celiac disease, they increase the transmembrane permeability that causes the activation of T cells driven by TNF alpha, and the autoantigen is transglutaminase. So the main problem in these kind of condition is that they have several extraintestinal manifestations, including infectious disease like TB or different GI infection, and also lymphoma. So can we have, can we predict this disease? Can we have specific therapeutic targets? Because as the last speaker, Professor Sanjeev was talking about, the gut is full of microbiota, and there are many different kind of competition happens throughout the lifetime, and people may get different infection because of increased transmembrane permeability. So when we are talking about different extraintestinal manifestations, we did a microarray study, and we looked at more than 200 genetic loci to find out genes which are implicated in celiac disease. I will not go through those graphs, just summarizing my finding, our finding that in juvenile and adult celiac disease, why we segregated juvenile, childhood, because childhood celiac disease has much more of genetic component, because CD is a complex disease where genetic environment, genetic component and environmental players play a role together. So in childhood genetic, they have a much more genetic component rather than adult celiac disease. But when you look into the data, we found, don't go to these statistical values, we found that ANK3, ankylin 3, that codes ankylin G protein, has come up in a very good way. Look at the significance level, -7 and -5. Is associated with the disease condition. So then the question is, this gene is known for neuropsychiatric condition for last 2 or 3 decades, and their role in neuropsychiatric conditions are very well explained in case of bipolar disease, schizophrenia, anxiety, major depressive disorders, and several other kind of conditions where ANK3 is very much implicated. So when we find out this gene in celiac disease, like an autoimmune disease condition, first we wanted to look at how this axis is working, how this particular protein, which is actually present in myelin sheath and implicated in different psychiatric condition, how it plays role in celiac disease. So are those individuals who are having neuropsychiatric conditions are prone to celiac disease, or individuals who are having celiac disease are prone to neuropsychiatric conditions? So this question, and we start answering this thing. When you look into the published reports, we found that there are several reports that says that there are psychiatric conditions are one of the comorbid conditions in celiac disease, and in our data set we have found more than 30% subjects having celiac disease are developing neuropsychiatric conditions over the time. And then we gone ahead with our data. We did a meta-analysis with existing reports, and just look at these p-values that we have found that these are the 6 different neuropsychiatric conditions which are already published, and with our data, we found that they are showing significant co-occurrence among celiac disease patients. So, what is the molecule and how these two diseases actually co-occur? Can we have a specific drugs? Because for autoimmune disease there are no specific drugs. Mainly we target T cells, mainly we target to suppress immune cells down. We give anti-inflammatory drugs like steroidal drugs. There are no specific drugs available. So can we find out a target that can handle both these disease conditions? Then we looked into the published papers, our own data. We found these are the 14 genetic loci across the genome. So there are not only one ANK3 that we have found. These are the 14 different loci which are common between neuropsychiatric conditions, schizophrenia, bipolar, anxiety, and depression, and they are common with celiac disease. So these are the chromosomes, genomic location, and the genes. Mind you, this HLA-DQ A1 and DQB1 are principal diagnostic markers for celiac disease as well. So when we diagnose celiac disease, doctor asks for HLA-DQ typing. So HLA-DQ2, HLA-DQ2.5, and HLA-DQ8, they comprises almost 95% of celiac disease patients. So if there are 100 patients, 95% are positive for HLA-DQ1 and DQB1. And surprisingly, these two genes are also common in these four conditions. And our target gene, ANK3, ankyrin G, that codes for ankyrin G, are also now shared between these two disease conditions. This thing is basically present in schizophrenia and bipolar disease. So these two locus actually gives antigen presentation and adenine junction. Then to find out what are the other genes out there in pathways and etc., we did a transcriptome study. So in our discovery cohort, we had a very small sample size, 3 CD patients and 4 non-CD controls. So these are not healthy controls. They have certain issues, so that's why they gone to the doctor. They perform biopsies and we got the sample with ethical permission. So those controls are basically suffering from dyspepsia. So those are not immune disorders, not infectious disease. Those are simply gastric ulceration. So these controls are not completely without inflammation, but obviously they are not, as depressive like celiac disease. So and these 15 and 21 green colored numbers are basically more replication cohort, but we tested using real-time PCR. So we found that 135 genes are downregulated and 788 genes are upregulated. I am not presenting the whole data, but for ankyrin G and CTH, that is E-cadherin, they also came up as a very significant gene which are downregulated in celiac disease patients. So what we are suspecting after genetic study, literature search, meta-analysis, we found that in our study, these two genes are downregulated. And they are also implicated in multiple pathways. I am not telling you all the stories, but these two pathways like mucosal membrane channels and synaptic membrane maintenance, these two are the most top two significant pathways that has come up in the pathway study using CAPE and gene ontology databases. So that's fine. Then we had a biopsy sample size from DMC Ludhiana. Then we did for immunohistochemistry with the patient samples and non-CD controls. For this ISC, we used 15 CD patients and 12 non-CD controls. We stained them for ankyrin G and E-cadherin and beta spectrin. They are actually interlinking 3 cytoskeletal components. So ankyrin G and beta spectrin are cellular component, and E-cadherin that is extracellular and transmembrane protein. So we found that in all the samples, 12 and 15, though the staining patterns are a little bit different because it's a complex disease, not all the genes are always involved. But when we found out a pattern that there is a significant downregulation of ankyrin G in the ISC samples. These are normal HE stain, just to show the degradation of microvilli structure. We did not find any significant change in the pattern and the intensity of beta spectrum. When we did real-time PCR evaluation on 25 and 23 samples, we found significant variation in ankyrin G, But for E-cadherin, the p-value was not significant, maybe the sample size, maybe the heterogeneity in the patient samples. But for N-cadherin G, we have found significant results. That indeed there is a significant variation. Part of those samples was also included in the transcriptome study. Then we went ahead to find out what happens if we downregulate ANKG. What happens in that gut tissue? So we used IEC6 cell line and we downregulated the ANKG with shRNA construct, and we gave 2 induction to actually mimic celiac disease-like condition in vitro. One is pepsin-trypsin digested gliding, that's the principal antigen for celiac disease, and TNF-alpha, which is a pro-inflammatory molecule, also implicated in celiac disease. We standardized these 2 treatment doses after NTT assay. I'm not showing the graph here. So when we did this study, so this first graph is untreated control. Then we gave upper panel, I think it's not visible clearly. This is N-cadherin, E-cadherin, beta spectrum. So these are the 3 N-cadherin, E-cadherin, beta spectrum. This is for immune expression level, and we found that when we give these PT glidin and TNF-alpha, all the gene expression comes down, and this come down is significant. This downregulation is significant. But when we give TNF-alpha, upper panel is for glidin. When we give glidin, the downregulation of N-cadherin and E-cadherin is quite significant. So if you compare these 2 graphs, you can see there is a significant downregulation, but for beta spectrum, the downregulation is not significant, the p-value is not significant. Same thing happens for TNF-alpha as well. This is the Western blot. You can see the faint bands of NKG and E-cadherin in different treatment condition. This is control, untreated, untransfected. This is knockdown plus glidin. This is untransfected plus glidin. Knockdown plus TNF-alpha, untransfected and TNF-alpha treatment. And this is the densitometric data to show the level of significance. So basically what we found is that PT glidin and TNF-alpha both downregulate NKG and E-cadherin, not beta spectrum. But when we downregulate, when we silence NKG, the downregulation is significant. So that means individual subjects who are having a mutation in the promoter sequence that leads to downregulation of NKG are more susceptible to react against glidin and upregulation of TNF-alpha. Now glidin is antigen for CD, and TNF-alpha upregulation can happen due to any infection condition or any chronic disease condition in the gut, maybe different microbiota interactions. So the subject who are having ANKG downregulation, they are more susceptible. Then we tested our model for transepithelial electoral resistance, TAR values. We found that in ANKG knockdown, there is a significant down of TAR. And then when we treated untransfected, transfected, KB is transfected one, we found that in all the cases when we added TNF-alpha and glidin, the TAR value is decreasing significantly. So when there is a downregulation, that means membrane barrier is now disrupted. So if you remove ANKG, the membrane barrier is getting disrupted. Second, we did study with dextran and FITC assay with 4 kilodalton molecular weight to check the transmembrane permeability. That means if there are 2 epithelial cells, what is the permeability between the 2 cells? Then we found out that with treatment the permeability is increasing, though these values not significant. These values not significant, upper value are significant. Then we have looked into the cellular morphology, normal light microscope, phase contrast. We found that glidin and TNF-alpha is changing the morphology of the cell, but in case of N-cadherin knockdown cells, the morphology change is very, very drastic. Just to look deeply into the data, then we did immunostudy, microscopy, and this is simple ANK and E-cadherin. in unconstricted cells, because we have seen that in unconstricted cells as well, they are down-regulating ANKG and E-cadherin. And we found that the distribution pattern of ankyrin is changing when treated with glidin and TNF alpha. There is more accumulation of the protein on the nucleus. Then for E-cadherin, there is a breakdown of cellular membrane. I can't say breakdown, but obviously mislocalization. The localization of E-cadherin is now changing. It is coming more towards the nucleus from the periphery of the cell. Then we did this confocal study. These are the unconstricted cells, these are the constricted cells. You can see the top picture where cells are unconstricted, and the picture is showing a very intact E-cadherin structure and good cellular morphology. But when you look to the bottom panel, which are constricted cells where ANKG is down-regulated, you can see the cellular morphology is changing, and also the mislocalization of E-cadherin due to knockdown and the treatment of glidin and TNF alpha is causing the mislocalization that is changing the cellular structure. So that says that the function of ankyrin G in myelin is well established, well known. But for celiac disease, it's an autoimmune disease. Here the function was not known clearly. So a person who are suffering or having a lower expression of ankyrin G due to any genetic polymorphism, that we have also found out, two genetic polymorphism for ankyrin G, which is causing down-regulation of the gene, and this may happen in the gut. Mislocalization of E-cadherin from the membrane, and most probably, we are not sure, we will be doing the study, that they are actually transporting those E-cadherin near to the nucleus, maybe. So that's how this is the scenario that happens. So maybe this E-cadherin and ankyrin is basically connecting the gut and brain axis, where there are many studies. Even Dr. Sanjeev was saying that patients there are neuropsychiatric manifestations. Well, so just summarizing this thing, this is the ankyrin, this is beta-stactin, and this is E-cadherin. That's the membrane structure. This is the total protein of ankyrin G, and this is how we are proposing the thing may be happening, that due to lower expression of ankyrin G, the internalization or the mislocalization of E-cadherin that happens, and with the induction of TNF alpha and beta glidin protein, there may be disruption of cellular membranes, and that may lead to celiac disease condition. So that's the first in vitro validation of non-immune genes in celiac disease. Other studies have used immune gene function to see how immune disease happens. But here we have presented a work where non-immune cytoskeletal gene can promote immune disease condition in genetically susceptible individuals. That means patient having IL23 positive. I would like to thank Professor Thelma and Professor Siska for the facilitating us for this in-the-chip study. We are part of International Celiac Disease Consortium, and then Dr. Ajit Sood and Vandana Mindra for providing us the sample from Ludhiana, Dr. Siva and Dr. Somesh to help us in cloning and transmembrane assay, and funding support from SAR, DST FIST and Central University of Punjab. And these are my 4 students. Just previous week, Nidhi, my first PhD student, just presented this work, because most of the work is from her PhD thesis. She just given the pre-PhD seminar, and most of the slides I have taken from her, and they are my next 3 students who are working on celiac disease and vasculitis. Thank you.
What is the clinical relevance of
There are many celiac disease patients where they are not confirmed by biopsy, because biopsy can see there is a degraded villi structure. But in present time, the phenotypes are bit different. There is intact villi structure, but patients are having high TTG level and HLA positive. So they are searching for different other molecules that may be used as a diagnostic marker. But if we look into the ANKG and how they are destabilizing the membrane, we can synthesize or we can
polymorphism in every patient, or do you find multiple places.
These are single nucleotide polymorphisms.
Yeah.
And these are the odds ratio. This is very high. Odds ratio generally more than 1 is very high in number. So we found that these two SNPs are strong LD with promoter. LD means linkage. So they have a promoter polymorphism, and these two polymorphism are inclonic. But they are in LD with SNP which are in promoter sequence. And when we look into the EQTL, expression quantitative trait locus, we found that this polymorphism is altering the expression of ANK3. So these are the two polymorphisms that we have identified, but these two are not causal. Causal may be the promoter one, which is downregulated by ANK3.
All right, thank you.
When we talk about E-cadherin, but at the time of junction between epithelial, there are many other junction proteins also present. E-cadherin alone is that much enough to have a breakage in that, or others are also downregulated or affected?
Yeah. So that maybe can be answered by this particular slide, because there are tight junction, gap junctions, and adherent junction. 3 types of junctions are present between two columnar epithelial cells in the small intestine. So when we knock down ANK3 and downregulation of E-cadherin happens, we can find there is a change in transepithelial voltage, right? But there is no significant difference in the molecule that passes through the membrane. So we can see there is no significant value because there are maybe gap junctions and other junctions that is still helping them to get the cells little bit closer. So that's why these molecules are not passing through the membrane. So we found this is significant because this is altering the charge, but not this one. We are suspecting that charge will be altered because in neuronal cells as well, because of loss of NKG from the myelin cell, the neuronal transmission does not happen properly. That's the biology. So we are expecting this thing to be coming positive, but surprisingly we did not find any good result in this assay. So that says whatever you are saying, that this alone is not able to destabilize, but obviously that will allow maybe the infection to happen, that because gut microflora influence in celiac disease is very, very well known. There are many studies that have found out that microbial populations are
The
involved.
size of gluten is smaller than this dextron NYPC?
Size of gluten, because there are heterogeneous population. I mean to say type of gluten. So we say like 22 mar, 33 mar, 23 mar. So there are multiple sizes of gluten molecule that transpass those membrane barrier. So depending on with what size, because the smallest size is 10 or 9 mar. 9 mar. Okay, so all of them are having an immunogenic portion of gluten that induces HLA through T cells. So yes, that's it. Thank you.
Thank you, Dr. Sabyasachi.
Thank you.
Thank you. I and my co-chair, Professor Lamita Gupta, would like to thank all the